Hello, everyone. We need to dig into question 1I, topic 1, the endosymbiotic theory. So let's see what we have here. It says 1I, the origin of eukaryotic cells can be explained by the endosymbiotic theory. It's a ton of vocabulary. So first things first, let's figure out what they're talking about when they say prokaryote and eukaryote. So when you look at cells, there's two major cell types, okay? And they're all based on the nucleus or the lack or absence of a nucleus. So there's two cell types. There's the prokaryotic cell, or the prokaryote, and these are simply the bacteria. And there's the eukaryote, and these are plants, animals, fungus, and protozoans. Protozoans are single-celled organisms that live in water. So there's two cell types, procs and eucs, prokaryotes and eukaryotes. Prokaryotes are the bacteria, no nucleus. The only organelle they have are ribosomes. And we'll talk more about them when we get into topic 2. And then there's the eukaryotes. These are the organisms that have rough ER, smooth ER, Golgi body, vesicles, vacuoles, nucleus, all of what we call the membrane-bound organelles. So when you look at a bacteria is pretty simple compared to a plant or animal cell. So bacteria, right? You have a plasma membrane. It may or may not have a cell wall. And since there's no nucleus, its DNA is just attached to the plasma membrane like this. And again, there's a lot more detail that we'll talk about later in Core 2. And then the only organelle that floats around out here in the cytoplasm are ribosomes. And it's the job of the ribosome, if you recall gene expression, transcription, and translation. During translation, it's the ribosome's job to link the amino acids together to make proteins. So of course, bacteria have to have ribosomes because bacteria have to make proteins. And then you may or may not have a flagella. Okay, it's a very basic bacteria. Now, when you look at plant cells, notice that I'm drawing it much, much bigger. Actually, the ratio here is about 10 to 1. So this plant cell should really be about this big, all right? But I don't have the space. So since this is a plant cell, you have a nice organized nucleus. That's why, by definition, this plant cell is a eukaryotic cell. And inside of that nucleus, you of course have DNA. And then floating around here in the cytoplasm, these are ribosomes. I'll shorten that down to ribose, right? Now, a major difference between plants and animals and bacteria is that plants have what we call the power plant of the cell, okay? This is an organelle called the mitochondrion. And it's called the power plant of the cell because inside of this cell is where the majority of the ATP, or the energy for the cell, is made. Okay? Now, plants are cool because then they have another organelle with little discs on the inside that we'll talk about in Core 8 next year when you guys are seniors. And this organelle, since it's green, is called the chloroplast. Okay? Now, what makes the mitochondrion and chloroplast so unique is that they have their own DNA. It gets better. They have their own ribosomes. They go through their own transcription and translation in here, separate from the entire cell. Now, bacteria reproduce by a process called binary fission. And we basically talk about this in high school. You'll get this more in college. And guess what? Mitochondrion and chloroplasts both reproduce by a process that's really similar to binary fission. That should be an S, by the way. There we go. Okay, so it's almost like mitochondrion and chloroplasts are like bacteria. In fact, they are extremely similar to bacteria. And if you look at an animal cell, so here's your animal cell, same idea. It's a eukaryote, so a nice organized nucleus with DNA, okay? Of course, ribosomes, so it can synthesize proteins. So there's some ribosomes, okay? Now, of course, since this is an animal cell, let me get this up so that you can see it. Since this is an animal cell, there's no chloroplasts. We don't have the ability to photosynthesize. I wish, wouldn't that be cool, right? But we, of course, have, as animals, we have that same power plant organelle. We have those mitochondria. All eukaryotes have mitochondria. And you now know that mitochondria have their own DNA, right? They have their own ribosomes. They reproduce by a process that's real similar to binary fission. So both types of eukaryotes have organelles that look a lot like bacteria. Okay, so there's something going on here where we have these bacteria-like cells living inside of larger cells, thus the endosymbiotic theory. Okay, so that's what 1I is trying to explain. Why, in the fossil record, do we have about 2 to 3 billion years of bacteria, and then in about the last billion years, we start getting eukaryotes? And how did these other cells that look a lot like prokaryotes, look a lot like bacteria, start to work in concert with, symbiotically, or through symbiosis, thus endo, inside, symbiosis, okay, working together but on the inside. So what 1I is really all about is, what is the origin of the eukaryotic cell? Because Miller and Urey showed us how you can make prokaryotic cells, how you can take some methane and some heat and some water and some ammonia and some lightning, and you can start making macromolecules. You can start forming the foundation of the original cells, prokaryotes, okay? So originally in the fossil record, you have prokaryotes. And here's what a lot of people don't know. About 3/4 of the fossil record, okay, about 2 to 3 billion years of rock is nothing but bacteria. We have billions of years of fossilized bacteria. Everybody talks about T-Rex and Triceratops and velociraptors. Don't get me wrong, beautiful animals, right? But the majority of the fossil record is actually bacteria, okay? So then what starts happening in step 2 is you start getting infolding or invagination of the plasma membrane. And of course, we'll draw pictures here. Actually, we will use the textbook pictures because their drawings are just a little bit better than mine. So you have the infolding of the plasma membrane, right? And what this starts to form is the nuclear envelope. Now, if these terms sound familiar because of regular bio, great. If not, and you're thinking, organelles, mitochondria, and chloroplast, nucleus, we will get back to these in topic 2. Don't worry, all right? So this is the nuclear envelope or the nucleus. So that's how the nucleus was formed, is the plasma membrane started to invaginate in or started to infold, right? Then you have step 3. Step 3 is then more infolding, or if you prefer invagination, fine with me, more infolding of the plasma membrane. And this gave rise to your organelles. And you say, like what? Oh my gosh, rough endoplasmic reticulum, or rough ER, smooth ER, vesicles, vacuoles, be sure I spell it right, and so on. So as the plasma membrane started to infold, this gave certain cells an advantage over cells that didn't do the infolding, right? They were more efficient at transport. Here we go again with surface area to volume ratio. So if you have 2 cells living side by side, and one cell has this infolding and is more efficient at transport, this cell will then be more efficient. It will be more successful, right? So You then get the formation of organelles, and you say, well, how do we know this? Fossilized cells. It's stunning. You have to look this up. All right. Then step 4, you have engulfing. Okay, to engulf, to take in. It's an interesting term. Engulfing of, and here comes some vocabulary, an autotrophic prokaryote. Now, I never want you to be intimidated by vocabulary, ever, okay? We will break these words down into their Greek and Latin roots and explain them, okay? So, auto means self. Troph means to feed. So these are individuals that have the ability to feed themselves. They feed themselves through photosynthesis. So an autotroph makes its own food through the process of photosynthesis. Okay? What they're really talking about and what this became in eukaryotes is the chloroplast. What the modern chloroplast, what that used to be, is it used to be an independent organism. Okay? It used to be an autotrophic prokaryote by itself. That's why chloroplasts have their own DNA. They have their own ribosomes. They go through their own gene expression. They reproduce separate from the larger cell they now live inside of. Okay? So chloroplasts came from an autotrophic prokaryote that was taken in and then existed or functioned through symbiosis, thus the endosymbiotic theory. Right? Then you have step 5, which would then be the engulfing, not of an autotrophic prokaryote, but of a heterotrophic prokaryote. So hetero meaning other, troph meaning feeding. So these organisms feed on others. Okay, they get their energy externally and they take it in by consuming. Right. So these organisms require what we will call food, macromolecules. Right. Require food from other organisms. Which organelle are we talking about here? The mitochondria. This is where the mitochondrion came from. This is why, plural, mitochondria, modern mitochondria have their own DNA. They have their own ribosomes. They go through their own gene expression. Why? Because they used to be their own organism. So the endosymbiotic theory is a 5-step process. You start with a prokaryote. You start to invaginate or infold the plasma membrane. That leads to the nucleus. That leads to the membrane-bound organelles. You then take in an autotrophic prokaryote. That leads to the formation of the chloroplast. You then take in the heterotrophic prokaryote, and that leads to the mitochondria. It's 5 steps. Okay. So then we then tackle this image here. So here's the Miller-Urey image that I spun for you. I spun this one too, trying to make every inch, every millimeter of this handout count. So what do you see on the top here? This is a prokaryote. And again, the majority of the fossil record, in fact, 3/4 of the fossil record is bacteria. It's prokaryotes. We have billions of years of fossilized prokaryotes. All right. So 2 to 3 billion with a B, billion years of prokaryotes, just layer after layer after layer after layer. Okay. You then get your infolding here. You get your infolding of the plasma membrane. And what does this start to form? This starts to form the nucleus. This gives the cell an advantage in transport, makes it more efficient, gives it an evolutionary advantage. You then get more infolding of the plasma membrane. This leads to the formation of other organelles, rough ER, smooth ER, Golgi body, vesicles, vacuoles, okay? Here's the engulfing process, okay? This is the mitochondrion here. So this is your heterotrophic prokaryote here, right? Which eventually evolves into the mitochondrion, right? So this original eukaryote cell has the mitochondrion. Then this splits off into two cell types. This cell type keeps the mitochondrion, but doesn't get the chloroplast. This is where animal cells come from. This is the modern animal cell, okay? Whereas there was then a second engulfing process of the autotrophic prokaryote. This is the chloroplast. This is where the chloroplast came from. And then with the symbiosis between the chloroplast and the cell, this is where modern plant cells came from. And how do we know this? Because it's in the fossil record. It's in the rocks. It's amazing, right? So this is question 1I in topic 1. Enjoy.